Hello, everyone. Welcome to my Biocool classes. Today's topic is lipids. So we will discuss about lipids today. So lipid is an important part of the biomolecules. Okay. So what are lipids? So lipids are a diverse group of compounds. So if you look at lipids, so they are a diverse group of compounds. And one important factor about lipids is they do not mix well with water. Lipids, they do not mix well with water. So this is very, very important concept about lipids. Okay. So this is something which all lipids, they share, that you will not find lipid which dissolves in water. So lipids, they don't mix with water. If you look at the component of a lipid, what does a lipid made up of? A lipid is mainly consist of carbon and hydrogen atoms. So carbon and hydrogen based. Now we have learned before in the biomolecule classes that carbon and hydrogen are known as the hydrocarbons, right? So when I say carbon and hydrogen, that's basically it's called hydrocarbon. Hydrocarbon. And any biomolecule is basically a carbon-based compound. Is a carbon-based compound. Any biomolecule is a carbon-based compound. So we need to remember these concepts. And lipids are something which are hydrophobic. That means water bearing. In general, as I said, because they do not mix well with water. So they're basically hydrophobic, they're water bearing. Okay. The concept of this, that lipids don't mix with water, we can see in an unshaken bottle of salad dressing. So if we look at an unshaken bottle of salad dressing, so basically the oil, which is the lipid, it separates from the vinegar. Okay, if you're looking at an unshaken bottle of salad dressing, so we're looking at the oil is the oil is a type of lipid. It separates from the vinegar, okay, which is mostly, the vinegar is mainly the water. Okay, so basically the vinegar is the water and the oil is basically the lipid, and it separates in an unshaken bottle of salad dressing. So if you take the salad dressing, if you keep it just like that for a long time, then what we see is that the oil, it separates from the vinegar. So we can see a picture of that. So this is basically the salad dressing. So you can see, if you look at this, if we study this picture, then we can see that this is a salad. So this is the salad dressing bottle, okay, and we can see that there is actually air at the top, and then these brown things, they represent the oil. So basically olive oil. And then the vinegar is the blue thing, which is basically the vinegar is actually the water part. Okay, so we can say the vinegar is the water part. Okay, and then this cream colored thing, this is basically the solids. So this is a shaken bottle. This is a shaken bottle. That means everything is mixed here, homogeneously mixed. Now we keep this bottle for a long time. So this is an unshaken bottle. So this is the unshaken bottle. And this is the unshaken bottle. And this unshaken bottle, you can see how the oil is separating from the lower vinegar. So this is the oil, and this is the vinegar, which I said is the basically the water. Okay, this is basically the water is the vinegar. So this shows how lipids, they actually, this is the common property of lipids, that how they do not mix with water. So to understand that, we can see a bottle of salad dressing where the oil is separating from the water, which is the vinegar. Okay. Next, we will learn the structure of lipids. That's very, very important. So in the biomolecule chapter, it's very important to understand the structure of these lipids, how they look like. Okay. So now, if you see that lipids are, they differ from carbohydrates and proteins and nucleic acids. Okay, they're neither huge macromolecules. This is very, very important. If you look at carbohydrates, if you look at nucleic acids, they are huge macromolecules, right? Carbohydrates are the polysaccharides. So these are basically the carbohydrates are the polysaccharides, right? They're the polysaccharides, carbohydrates, and we have the nucleic acids are the polynucleotides. Polynucleotides example is DNA, right? RNA, right? Nucleic acid. So they're huge. DNA is 2 meters long, right? But if you compare the carbohydrates and nucleic acids and also the protein, which is the polypeptide, right? If you compare them with the lipids are not huge macromolecules. So they are not polymers built from similar monomers. So lipids are not as huge as protein. They're not as huge as carbohydrates. Neither they're as huge as the nucleic acids. So this is one thing we need to understand. Then another thing about the lipids is uh, a fat is a large lipid. What is a fat? We have heard about the word fat. So fat is basically, it's a large lipid, and it is made up of two kinds of smaller molecules. Very important. This is very important. Fat is basically a large lipid. Okay, what is a fat? It's a large lipid. Okay, and it is made up of two kinds of molecules. One is the glycerol, and one is the fatty acid. So this is something we need to understand, that basically a lipid is made up of glycerol and a fatty acid. What is a glycerol? So let's try to understand what is a glycerol. So when I draw, so as I said, that a lipid like a fat, which is a large lipid, it is made up of two components. We have glycerol and fatty acid. Now, a glycerol is basically made up of three carbons, and each carbon has a hydroxyl group, and rest are all hydrogen, because each of these carbon, they need 4 bonds, right? So 1 2 3 4, 1 2 3 4, 1 2 3 4, right? So this is basically the structure of a glycerol. What are fatty acids? Fatty acids are long chain hydrocarbons. Hydrocarbons means carbon hydrogen. So it says CH3, then like that. You see that? That's how they look like. Each dot represents a carbon. And so we can see each carbon has 1, 2, right? And then there are 2 bonds which are there sharing with which I'm going to draw in black. So these two bonds they are sharing with the hydrogen. Okay, so there is again hydrogen. Each of these dots represent a carbon. So basically, if I draw this without this kind of stick and dot structure, without the stick and dot structure, it looks like CH3, CH2, CH2, CH2. Each of these dot represents CH2. Each of these dot represents CH2. So this is basically a fatty acid. It's a long chain hydrocarbon. So fatty acid is a long chain hydrocarbon. What is hydrocarbon? Carbon and hydrogen. Carbon, right? So this is like CH3, this is CH2, this is CH2, this is CH2, and so on. And these fatty acids, they can be as long as they can have 14 carbon, they can have 16 carbon, they can have 18 carbon. Okay, so they can, and when you count the carbon, you also count the final carboxylic group. This is the final carboxylic group. Okay, so we can, in this green I am drawing, so this is the carboxylic group. So basically you count this carbon also when you count the total number of carbons of a fatty acid. Okay, so what are fatty acids? Fatty acids are long chain hydrocarbons. What is hydrocarbon? Hydrocarbon is made up of carbon and hydrogen. Okay, and okay, okay, next. So coming back to the concept of glycerol and fatty acids, let's look at a picture. So now this is the picture of a lipid. So I need to move this down here. Okay, this is a picture of a lipid. And you can see this orange box is basically the, so this is the glycerol, as I said. And look at the glycerol. This is basically a 3 carbon, right? And there are hydroxyl group attached to each of these carbon. All right, and this is a fatty acid. Okay, they are actually, if we count it, there are 16 carbon here. Total there are 16 carbon, and we count this carboxylic acid carbon also, right? So this is a kind of so we call this fatty acid as, so we can see this picture, we call this as the palmitic acid. Okay, so this is called the palmitic acid, which has 16 carbon. Okay, this is a palmitic acid. You can see there are 16, total 16 carbon. And again, this is a hydrocarbon with carbon and hydrogen. Okay, and look, this is the carboxylic acid, right? So this is basically the 16th carbon. This is the first carbon, and this is a long chain fatty acid. Okay, it's called the palmitic acid. The formula is C16H32O2. Make sense? As the carbon number increases, so we have, so now when we look at the different types of long chain fatty acids, so we have palmitic acid, which has 16, right? We have stearic acid, which has 18. You see C18. So why it is saying C18 0? What does the 0 mean? 0 means it has no double bonds. No double bonds. You see this carbon and hydrogen chain. So each of these, remember, this is the carbon, this is the carbon, this is the carbon, this is the carbon. Okay, all these are carbon. Like that, there are actually 8 18 carbons there, and you will not see any double bond. That's why it's C18-0, right? Next we go is the myristic acid, which is C14. So C14 is myristic acid. Then we have the palmitic acid. Then we have the stearic acid. Okay, so you see the carbon numbers are getting higher. We come to something called the oleic acid. Here we are seeing a double bond here. Okay, so these three, palmitic, stearic, and myristic acid, so they are called the saturated fatty acids. Okay, so we call saturated fatty acid. This is called saturated fatty acid because there are because all the carbon bonds are satisfied, right? So saturated fatty acids, all the carbon bonds are satisfied. You see, not a single carbon is not satisfied. When you look at oleic acid, which has a double bond here, it has a double bond here, right? So this is called a unsaturated Fa, unsaturated fatty acid, right? Also, when we look at the picture of linoleic acid, linoleic acid has a double bond here, a double bond here, right? So you see the word C18-1, that means oleic acid has total 18 carbon, right? It has total of 18 carbon, right? And there is one double bond. That's why it's called C18 ratio 1. Okay, that's how we write it. When we go to linoleic acid, we can see it's C18. Again, it has 18 carbon and ratio 2. That means there are two double bonds. Here is double bond, here is double bond, right? So C18. So these are both are unsaturated fatty acids, and these three are saturated fatty acids. Okay, so that is something we need to understand. Next. Now when we come back to this figure, now everybody can understand what this figure means. So we have a glycerol. As I said, a fat or a lipid is made up of two components. One is the glycerol and one is the fatty acid, right? And in this case, this is the palmitic acid because it has 16 carbon, right? Palmitic acid. And you can see now very, very interesting thing. There is something called the dehydration reaction that is happening when the glycerol, which is called glycerol moiety, right? When the glycerol moiety is joining with the fatty acid, you see a water is getting released, right? So how the water is getting released? So here one of the hydrogen from the glycerol and the hydroxyl of the carboxylic group, right? So this is basically the carboxylic group and this is the hydroxyl group. So you can see that the hydrogen from the glycerol hydroxyl group and the carboxylic acid, the hydroxyl group, they're getting out by a dehydration reaction, and then this fatty acid, this long chain palmitic acid chain, is joining with the glycerol, right? So this is basically a mono acyl glycerol. It's called a monoacyl glycerol. Can you see that? It's a monoacyl because there is one chain fatty acid, right? So here, so this is a monoacyl glycerol and this is a triacyl glycerol. This is a triacyl glycerol, right? So mono means one fatty acid. So if they would have been two of these, it would have been diacyl glycerol, right? So this is actually monoacyl diacyl glycerol. Okay, so diacyl glycerol, if there would have been two of these, right? If there would have been two of these diacyl, if there are three, it's called triacyl glycerol. So triacyl glycerol is basically called a fat. Okay, so fat has three of these fatty acid, long chain fatty acid chains. So when you go back to one of my notes, you can see that linking of 3 fatty acids to the glycerol produces a fat, right? So 3 fatty acid produces the fat. Okay. Now, synonym of fat. What is the synonym of fat? Synonym of fat is triglycerides. Okay, so these words are very important. What is a fat? Fat is basically when there are 3 fatty acids joined to the glycerol by dehydration reaction. That's called a fat. Okay. Fat is also called triglycerides because there are 3 fatty acids. Okay, and we see on the food labels, right? When we order for a medical test, we also see that our triglyceride level has gone high, right? So that means we are having a lot of consuming a lot of fat in the body, right? Body has lots of fat, so that's called the triglyceride level gone high, right? So also in the food level, you see triglyceride, okay? So on the medical test for fat in the blood, right, we see triglyceride level going high, okay? So that is something which is, so what is fat? What we learned, fat is basically triglyceride. What else we learned? Lipid. So lipid. So here's the thing. Here's one thing we need to understand before it gets complicated. So basically, when we say a lipid is made up of a glycerol and fatty acid. Now a lipid can have 1 fatty acid, 2 fatty acid, 3 fatty acids. So a lipid can be monoacyl glycerol, diacyl glycerol, or triacyl glycerol. But when we say fat is a specific kind of lipid which has glycerol joined with, so with a different color, joined with 3, look here, this is 1 fatty acid joined with the glycerol by dehydration reaction. This is another fatty acid, right? This is the second fatty acid, and then we have the third fatty acid, which is joined with the glycerol. So now this triacyl glycerol is basically what is called a fat, right? So fat is a type of lipid which has 3 fatty acid, long chain fatty acid chain joined to the glycerol moiety, right? And we know what is the glycerol. It is basically having 3 hydroxyl group attached to the hydrocarbon backbone. Okay. All right. So next we go to so we learned about these things. Now, saturated and unsaturated fatty acid, already I've discussed what is a saturated, what is an unsaturated fatty acid. As I said, fatty acid could be saturated or it could be unsaturated, right? Based on the double bonds. Okay, so it can have 1 or more double bonds, just like given the example of, now we will go back to this figure and we can see that it can have either 1 double bond, just like in oleic acid, it is C18 ratio 1 double bond, or it can be linoleic acid, where we have 2 double bonds, which is C18 ratio 2, right? The ratio sign 2 because there are 2 double bonds, right? So unsaturated fatty acids can have 1 double bond or it can have 2 double bonds. Okay. Very, very important concept, very interesting concept also, I would rather say, that 1 or more double bonds in unsaturated fatty acids, it causes a kink or a bend. Because of the double bond, there is a bend. Now, again, when we go back to this picture, we do see that kink. Right, when we go back to this picture, we do see that, look, when you look at a saturated fatty acid, when you compare a saturated with an unsaturated, so let's look at this. So these are the saturated guys, okay? This is the saturated ones, which does not have any double bond. All the carbon valencies are satisfied, okay? Here, in case of the unsaturated ones, you see there is a kink. Can you see how bent it is? Like that, and then there's a bend, okay? Again, this comes like that, and then there's a bend. Wherever there is the double bond, there is this kind of bend, okay? So this bend has a huge impact on the physical property of the of this particular kind of lipid. So what it does, when you have a bend, when your lipid is bent, then it is very hard to pack it, right? Compared to that, if your lipid is straight chain, if there is no double bond, then it's straight chain. It's very easy to pack these fibers of straight chain. Okay, but if there is a kink, which is called the kink or the bend, so because of this kink or this bend, because of this bend, it becomes very, very difficult for the unsaturated fatty acids to be packed. And since they cannot be packed, they stay as liquid. If it could have been, if it could have been tightly packed, they would have been like solid, right? So here we have this concept. So here we have the concept. So the kink in the unsaturated fatty acid prevents fats containing them. That means fats which have unsaturation, which have double bonds. It prevents them from packing tightly together and solidifying at room temperature. You see, so these unsaturated fatty acids, they are basically liquid, just like we have corn oil, we have olive oil, we have vegetable oils, right? So they are all unsaturated fatty acids. But when you look at most of the animal fats, they are saturated. Okay, so the animal fats are saturated. What is the example of an animal fat? So an example of an animal fat will be butter and the beef fat, right? So beef fat, they are solid at room temperature, right? But when you look at corn oil, olive oil, vegetable oil, okay, and other types of vegetable oils, they are basically liquid because they're all having their double bond. They have the kink; it can't be packed. But the saturated fatty acids, they don't have any double bonds. They can be easily packed. So most of the animal fats, they are saturated, and so they are solids. Like beef fat is solid at room temperature. Okay, so now we'll try to understand the medical significance of uh, of uh, having unsaturation, and the medical significance of having saturation. So now we look at this on my left side. We have, these are all the different types of oils. We have the corn oil, we have the peanut oil. Okay, so these are all unsaturated fatty acids. So they are all unsaturated lipids, right? So we call them, because their fatty acids have double bonds. Okay, so these are all unsaturated. Okay, when you look at these oil, when you look at the beef fat and when you look at the butter, they are basically saturated. They can be packed closely. That's why they are solid at room temperature. Okay, very important concept. Now, very, very important is the concept of trans fat. What is trans fat? How trans fat increases, it causes a health risk, how it poses a risk for heart disease, right? Now, as I said, um, so here, when you see, um, so when you can pack fats, try to understand this concept. When you can pack fat very closely, which we see in the saturated fatty acids, then there is a tendency of forming plaques or deposits in arteries. Okay, so that means saturated fatty acids can easily form those kind of small globules, or they can form those kind of small plaques inside our arteries, right? So here, so when we go to, so let's say this is our arteries, right? This is our arteries, right? So now the probability or the possibility of a saturated fatty acids, so a saturated fatty acids or saturated fatty acids, they have a higher tendency. So since they can pack closely, right? Since they can pack closely, they can easily form they can form these deposits inside the arteries. They can form those small plaques which roam around in the blood vessels. Okay, this can lead to heart um, failure. Okay, right? Heart failure, because packing closely can lead to formation of plaque, right? Plaque means these kind of deposits, right? They form, which causes problem in the blood flow, right? This leads to increase in blood pressure, right? It can lead to cardiac arrest. Okay, so saturated fatty acid is bad, especially those which have trans fat. Okay, so what is trans fat? Let's try to understand this concept of trans fat. So trans fat is, let's say you have a unsaturated fatty acid, right, which has multiple double bonds, right? Just like we said, linoleic acid. Linoleic acid has two double bonds, right? But now you're hydrogenating the double bond. That means you're adding hydrogen and you're converting the double bonds into single bonds. You're saturating the unsaturated fatty acid, right? So in that case, that is called hydrogenated vegetable oil. That means the vegetable oil is an oil in the first place because it has double bonds. It can't pack. It has kinks. That's why it's liquid, right? So now you have hydrogenated. Now you're converting the double bonds into single bonds, right? And in that process, you're creating a trans configuration. So let's try to understand this thing. So this is might be a little bit difficult concept, but I'll try my best to explain this. So now what we have here, let's say this is a carbon, this is a double bond, right? Okay. And we have here another double bond, right? So this is basically the hydrocarbon chain. Now, here we have hydrogen, remember carbon has 1, 2, 3, 4, 1, 2, 3, 4, right? And here there is hydrogen, okay? Try to understand this concept. So we have a double bond here, we have a double bond here. This is the original unsaturated oil. So this is basically the oil which has, this is the oil which has these unsaturation. Okay. Now we hydrogenate. That means now we are adding hydrogen. We are adding hydrogen. We are adding hydrogen here. We are breaking this double bond. So when we do that, so now how do we draw the structure? So now C, this is the carbon, this is the hydrogen. So now this becomes CH2, right? And here the double bond still exists. So we have hydrogenated this part. So let's cover that in green. So this part has been hydrogenated. Okay, this part has been hydrogenated, right? This part, now look at the H. This H, both the H are on one side, right, of the carbon. Both these H are on one side, on the upper side, right? So this is called the cis configuration. This is called the cis configuration. Now, once we hydrogenate, once we add hydrogen, now let's see. One hydrogen remains on this side, another hydrogen goes on that side. Can you see that? So now let's go back and finish the rest of the carbon backbone, right? So this configuration, so now we just, so now this configuration is called the trans configuration. Okay, this is the trans configuration, right? This was the cis configuration when the hydrogen are on one side. This is the trans configuration. So this is called a hydrogenated vegetable oil, right? This is the vegetable oil and we are trying to hydrogenate. We're trying to add hydrogen, okay? So this is the hydrogenated vegetable oil. And this is a trans fat. This trans fats, they have a very high tendency of forming cardiovascular disease. Okay, so such a converted vegetable oil, a vegetable oil where we are adding hydrogen and we are converting the cis configuration to a trans configuration, has a very high chance of forming plaque, of forming arterial diseases, of forming cardiovascular diseases, and that can lead to heart failure. Okay, it can lead to heart failure. So this is, so now we go to some pictures to understand this concept. So this causes health disease. It can cause atherosclerosis. So we can see, look at this picture. This is basically, we are talking about this unsaturated, this is the linoleic acid, right? So linoleic acid has, so now when we look at this picture, as I said, the linoleic acid has two, it has two double bonds, right? Two unsaturation. There is one unsaturation, there is another unsaturation. Can you see that? This is before the hydrogenation process, right? And look at the hydrogen. The hydrogen is in the cis configuration. They're on same side of the carbon. So now we hydrogenate this. We add hydrogen. Now you can see this double bond is gone. This double bond is gone, right? Because we have added hydrogen here. All right. So this double bond is gone here. This second double bond is still there, but look at the hydrogen, it has shifted on the other side and it has taken the trans configuration. This is a very dangerous thing. This kind of hydrogenated vegetable oil has a high tendency, when you take this hydrogen trans fatty acids in through our food, they can cause blockage of our arteries and cause these kind of cardiovascular diseases, which is called the this is called the atherosclerosis, okay, right? Which is basically build up within the walls of the blood vessels. Okay, so this is basically so diet rich in saturated fatty acids and trans fats may contribute to cardiovascular disease by promoting the atherosclerosis. In this condition, lipid-containing deposits called plaques, they build within the walls of the blood vessels, reducing the blood flow. Okay, what is the solution? We should have lots of unsaturated fatty acids. Examples are the omega-3 fatty acids, okay, which are found in the nuts, which are found in the plant oils, found in the fatty fishes, okay. And so nuts, having plenty of nuts, having plant oils, plant-based oils, not animal-based oils, fatty fishes, okay. In fish, we can see lots of, so fish has an oil, right? Any idea that fish has an oil? From fish, there is oil coming out, and this is called omega-3 fatty acid, which is basically unsaturated fatty acid. It has those kinks, it can't be packed closely, so they remain as a liquid form, and they don't have the tendency of forming these kind of plaques, right? And they protect against the cardiovascular disease. Very, very important, okay? So now we know what is saturated fatty acid, what is an unsaturated, what is a kink, what is hydrogenation of vegetable oil, how that poses health risk, what is the trans configuration. So now here are some of the vegetable oil labels you can see, and you can see how this is a clearly, you see this label, it says partially hydrogenated soybean oil. So now when you read a label like that, you will understand what does this partially hydrogenation means, right? You can read this label, and you can also see how this here the trans fat level is already given here. Okay. um, so let me just